All right. Hello, everyone. Well, it's been a fairly eventful last few days. And, I just wanted to touch on this. As it's always the case, it's going to be interesting to see if and how science policy changes as a new administration comes into the White House. A new president was just inaugurated, in case you're unaware. Before the inauguration, we knew that there was going to be at least one pretty meaningful change. So the director of the Office of Science and Technology Policy, or the OSTP, has been elevated to being a cabinet level position. And so I'm not going to spend too much time on this, because there's plenty of places we can all go to get updates about our government and politics and stuff like that. But this is a pretty meaningful event for the influence of science and scientists in government policy, and here's why. So the Office of Science and Technology Policy, again, OSTP is how it's usually called. It hasn't been around for all that long. It was established in 1976, and it has a pretty broad purview with the capacity to influence decisions in the White House that influence everything from health and the environment to investments in research and technology and STEM education, but also things like economic development and national security. And, pretty much before 2018, you could fit every single scientist that was serving in Congress in my tiny Philadelphia apartment, pretty much. Right, so in 2018, there were more scientists and engineers who were added to the ranks, right, during that midterm election, and the specific numbers depend on what disciplines are considered to be technically science, like engineering or medicine or dentistry, that kind of thing. But it's still not a particularly strong representation within our elected government, right? So it's certainly good that the influence of an office like OSTP will be amplified within at least one branch of the federal government. And I'm pretty sure it's going to be the first time that OSTP will be headed up by a biomedical scientist rather than a physicist or some something like that, which might be slightly biased for fairly obvious reasons. But with the inception of the proliferation of platforms like CRISPR, the potential emergence of entirely new vaccine technologies, and the increased integration of technology into our biology, right, with whole brain computer interfaces, Neuralink, companies like that. And then also the prospect of antibiotic-resistant bacteria. I think having someone who's been at the cutting edge of biomedical science is just going to be prudent. All right, on to the topic for today. Oh, I should note, my name's Mclaughlin, and I have a PhD in neuroscience from the University of Pennsylvania here in sunny Philadelphia. Okay, so today I wanted to talk about a topic that's basically been sort of at a slow boil for the past several years, the microbiome. And so I know that sounds like a word that might be, somewhat reminiscent of a 90s movie, a thoroughly 90s movie. Or, if you're as nerdy as my parents were, perhaps you might think of the biosphere experiment, which I actually visited with my mom when I was a kid. And honestly, it was a super fun trip that we took while we were in Arizona. And it's just it's a super interesting effort, if not, somewhat sort of ambitious. But anyways, that's not what I want to talk about today. So when it comes to the microbiome, the biosphere is certainly related, right? While the biosphere, was devised with the goal of creating a self-contained, closed environment, again, that's the biosphere, containing essentially all the essential components of the various ecosystems that feed, each other on Earth, including biological components. The microbiome refers to all of the trillions of microbial organisms that inhabit your body. So in other words, you're a walking ecosystem for trillions of microbes. Just like cheetahs or penguins can only really survive in certain environments, the microbes that currently inhabit your body have flourished because you have cultivated specific conditions that enable them to, right? Or just as importantly, you've been exposed to environments that have sort of sculpted the conditions that favor certain microbes over others. And these are all going to be loose estimates, but one that's kind of fun is that the total gut microbiome weighs about 2 kilograms, which is a fair bit more than the entire human brain. And another fun quantification of the level of activity of the human microbiome is that while we have somewhere around 20,000 protein coding genes, the total number of genes encoded by the microbes that inhabit our gut present several million genes that are expressed. And so just by the way, the whole humans only have 20,000 genes figure has been around for quite a while now, but I think it's just worth acknowledging that this number really only refers to protein coding genes. The number of genetically relevant sequences in the human genome is considerably larger, but it doesn't come close to millions. But I've seen a figure of somewhere around like 45,000 genes that do something, whether it's encode a protein or have some other sort of regulatory purpose. But anyway, so the best metaphor, in my opinion, is seeing the human body like a coral reef, right? If you've never gone like snorkeling or scuba diving near a coral reef, I very highly recommend it. At least when we're on the other side of COVID-19, it's a transformative experience. And I actually wrote my college application essays based on my experience at a coral reef, having to throw up through my scuba regulator. But that's a story for another day. But so coral reefs are, incredibly complicated ecosystems, depending on where in the world the coral reef is and what's present in the environment. Different types of corals, anemones, and fish and stuff like that will be present. But just, as the fish and anemones are themselves living organisms, the coral itself is essentially a living organism. So it's a living organism that's colonized by other living organisms, and neither could survive. At least they couldn't flourish without the other, right? Without the presence of the other. And they've evolved together over hundreds of thousands, hundreds and hundreds of thousands of years. And so there's a similar type of relationship here, right? Just like fish and coral and so on have evolved together, so did primates and the microbes to which they're exposed. And the similarities don't stop there with this comparison. So there have been studies that explore how people's microbiomes vary according to geographic location. Right, so there have been like comparisons of people in across 3 different continents, like Europe, the United States, and China, and meaningful variability in microbiomes have been detected, despite the fact that we're all Homo sapiens, right? There have also been comparisons of the microbiomes of people in North and South America, and then between Europe and Africa, Korea and Japan, and then rural parts of Russia and China, and those studies have also found, variations in the microbiomes as a function of diet, geography, early life exposures, very important component, and genetics. And it's unclear which plays a sort of dominant role in determining microbiomes among those different criteria. But who cares, right? There are these little microbes that are thriving in various parts of your body, but it's not like you knew about them, right? And, one interesting thing to consider is that while all of our genomes are extremely similar, the relatively minuscule genomic differences that we all have give rise to like all of the phenotypic variation about which we care so much, right? And like by that, what I mean is that, just a little bit of genetic variation gives rise to everything from distinguishing like blonde hair from strawberry blonde hair, or like dirty blonde or brown or black, curly, wavy hair and all those types of things, right? We all care so much about that. And we're literally talking about like a minuscule fraction of the genome that gives rise to all of that variability in just one component of our appearance, right? Just our hair, but we care about it so much. So like fairly subtle genomic variability can give rise to pretty overt differences in phenotype and what we look like, basically. And if a mad scientist were to like alter just a few of those genes, your hair might go from wavy to straight or curly. Right, so imagine, there are these trillions of microbes living, growing, reproducing within various parts of your body at all times, whether you're awake, whether you're asleep, doesn't matter. And there's, a ton of different microbes, different ones that inhabit different parts of your body, meaning like different microbes will find your intestines hospitable, then will find like your armpits or your mouth or your face or your nose hospitable. Different environments, different microbes. So, just to give you sort of a broad sense of how much of the body is colonized by microbes, here's an image that sort of simplifies the prevalence of different populations that inhabit, different parts of the body. And I know it's almost impossible to see the names of these microbes, in which parts of the body that they inhabit. That's not really why I'm bringing this up. More, I just want to impress upon you that essentially every part of the body is colonized by microbes. And different parts of the body are colonized by different kinds of microbes. And so, of course, it's not like this is a new phenomenon, right? We didn't just like start growing microbes all over ourselves all of a sudden. We co-evolved with these microbes. They, adapted to the environments that are determined by, our biologies, while we adapted to the environments in which we were evolving, right? And so, as we were evolving, these microbes that inhabit various parts of our body were evolving as well. So, to be clear, these are microbes that you have no idea are there, right? When you're healthy and when you're sick, right? They're just your sort of fellow travelers. And you received many of them from your mother when you were born. Like literally the process of being born seeded your body with the various microbes that inhabited your mother. And then, the environment in which you grew up of course, influenced, which microbes were capable of thriving and which ones ended up just dying out, becoming outcompeted. So again, like, who cares? Over the past few years, biomedical scientists have found that there are specific patterns of microbes that inhabit the bodies of people who, confront various biomedical conditions. Dysbiosis, which is a term to mean like a problematic balance of the microbial populations that inhabit the body, has been pretty consistently associated with a pretty wide variety of diseases. And when I say like wide variety, we're talking like everything from diabetes and asthma to inflammatory bowel syndrome, or inflammatory bowel disease, and multiple sclerosis, right? And there's a bunch of other conditions that have been associated with like a specific, microbial population or cluster of microbial populations. But like central to the concern over changes in microbial populations is that, like, if we were to introduce a radical change in an environment in, the biosphere, once again, like changing the temperature of an Arctic environment to be more like an equatorial environment, radical changes in, the microbial populations of the body may produce substantial changes in host biology. And by host, of course, I mean the human, like you. And so, before we get too far into this discussion, I think it's worth acknowledging that this is still like fairly fresh scientific territory. And there's plenty of good science that's already been done, but it is still kind of a young field. But okay, so with that stipulated, biomedical scientists have been interested in understanding the relationships that we have with all these little fellow traveling microbes that inhabit our body, right? And but with an eye towards figuring out if there's a clear distinction between the types of microbes that inhabit the bodies of folks confronting various medical conditions and those that inhabit, the bodies of people who don't have any medical conditions. And there's quite a wide diversity of the microbes that inhabit the bodies of people who are completely free from disease, right, and are otherwise healthy. There's not like the healthy combination of microbes that's perfect for everyone. And as a result, it's been pretty thoroughly difficult to identify something like an optimal combination of microbes for, a human. But we got to start somewhere, right? And so, one goal has been to identify a kind of like core set of microbes that are essentially always present in the bodies of people who are free from disease. Like what appears to be the bare minimum that we always encounter in the bodies of people who don't suffer from any medical condition. But even that goal has been, incredibly difficult because the level of variability just among healthy people is pretty massive. So the effort evolved a little bit. Instead of just finding the sort of like bare minimum recipe of microbes, one of the dominant strategies became to identify what has been referred to as a functional core, meaning like a set of microbes that are complementary to various individuals, and would therefore, not necessarily be the same for everybody, right? Given our, just like we all have, different colored hair and eyes, and, we're different heights, we have different predispositions to diseases and so on, and we have, similar levels of variability in our metabolic systems. And so, because these microbes, particularly the ones that inhabit the gut, of course, perform all kinds of biochemical reactions while they're living within us, some people's metabolic systems may be better suited by a microbial population that's capable of generating some kind of like biochemical reaction than, another person who has, a variation in their metabolic system, right? So sort of like how, like, depending on the melanin content of a person's skin, different levels of sun exposure can be tolerated, right? Not enough melanin or insufficient melanin for an amount of sun exposure, you get a sunburn. So it's sort of similar, right? It's the same environmental stimulus, right? The sun. But, due to biological and genetic variation in humans, that same environmental stimulus is differentially tolerated, right? And so that's pretty much what we're talking about when we're looking for a like functional core of microbes inhabiting people's bodies. One microbe might be beneficial or more detrimental in one person than it is in another. And so, I don't love this figure. It's kind of busy. It's kind of busy and noisy down there, but it basically conveys what we're talking about here. So, if you compare the microbiomes of 3 people who are healthy, there are going to be all kinds of differences in the gut, in the skin, in the mouth, and so on, other parts of the body. But if you begin to notice that there are specific microbes that are particularly prevalent in all 3 of them, and they happen to be healthy, you might have identified something like a central functional core. Okay, so as noisy as that figure was, let's get it out. So you can see, like there's some consistent little shapes that are, consistent across those three different people, and then the, prevalent microbial taxa. That's what they're talking about. All right, let's get rid of that picture. Okay. So I have another figure that's going to be like equally inscrutable. Oh no, wait, I didn't add it. Here, okay, I'll do that right now. We'll do, here we go. Where are we? Here we go. It's gigantic. Okay, that's fine. Adapt, overcome, improvise. Okay, so sorry about that. So that past figure was pretty noisy. This one's probably just as inscrutable for most people, but just bear with me. It'll make sense. So imagine that yellow dot over there is on the right-hand side of the picture, in other words, is an infant that was just born, okay? It doesn't, spontaneously have its own microbiome, right? Its own unique collection of microbes, but very, very rapidly after being born, the infant becomes colonized by the microbes that inhabited its mother, particularly when infants aren't born via C-section. And there's this whole body of literature, by the way, and the evidence that the maternal microbiome colonizes a newborn is robust enough that many biomedical scientists now advocate that when an infant is born by C-section, the infant very rapidly be exposed to the microflora of their mother to get access, to get that process started, that colonization process started. It's kind of cool. But so anyway, so the infant is just born over here. Can I ask for a refund on some of my microbes? Yeah. An infant is just born, rapidly becomes colonized by whatever microbial populations are in the area, in the environment, and the majority of which from its mother, of course. And the microbes begin to flourish and colonize the body of the infant. And generally, the microbial populations that colonize the infant will tend to reflect those of healthy individuals. So it begins to move as that line begins to move towards a population that would be considered healthy. And then, as the baby begins to grow up, its microbial populations can sort of vacillate a little bit. As the baby begins, eating new foods, playing outside in the dirt and stuff, and like interacting with babies from other families and so on, microbial populations will begin to evolve a bit. It'll change slightly, right? And it'll just sort of like orbit that sort of healthy region towards the green healthy region towards the bottom there. The microbiome isn't static. In other words, it's evolving, and it's generally evolving in a broadly healthy way. And then all of a sudden, there's some kind of perturbation, okay? And the baby, gets sick, for example, and has to take antibiotics, or perhaps is just exposed to a particularly unhealthy microbe. Or its diet becomes rather unhealthy all of a sudden for whatever reason, right? That perturbs the baby's microbiome. That's represented by that white dot. So now the baby's at the white dot. So the baby is in a precarious state, okay? And, let's focus on the case where the baby, gets a bacterial infection and must be administered antibiotics. It's sort of like an atom bomb, right? Completely decimating the microbes that had colonized the baby, right? Eliminating the beneficial ones along, of course, with the pathogenic one that it just caught, that infected it. But then, that opens the door for the baby's body to be colonized by completely new sets of microbes. So, in that moment, a few things can happen, okay? Option one, first, the baby may just recover, over the course of the antibiotic treatment and then just get re-exposed to the microbes to which it had been exposed, for its entire life. And then basically return to that bottom green area, right? A healthy population of microbes recolonizes its body and everything is just hunky-dory, right? It's doing fine. Another possibility number two, the baby recovers from infection, but because its environment isn't anything like the one to which it was exposed when it was born, and in its infancy, it's colonized by a whole new set of microbes that share some features with the initial microbiome, but it also has a whole new population. But this whole new population is also essentially healthy, okay? And so, even though it's a departure from its initial healthy microbiome, it's still compatible with the baby, and therefore it's still healthy. But now the baby's up here in this green zone, in this upper green zone, but still in a green zone. A third possibility, less optimistic possibility, the baby recovers from the initial infection, right? And the antibiotic treatment, it works and everything. But because the antibiotics open the door to the baby being colonized by any kind of microbe, essentially, it ends up being colonized by microbes that are essentially unhealthy. since the beneficial microbes are no longer there to fight off the unhealthy ones and you know, take up all the nutrients available to them, and the unhealthy ones are capable of thriving, and you know, and because those unhealthy microbes aren't compatible with the baby's, you know, own biology, it drives them towards a predisposition to disease states. So now that third possibility is this red zone, right? So that's the basic idea of you know, how we develop and evolve with microbes inhabiting our bodies. And I know that was like a fairly laborious intro, but I think it's useful to understand and know for what's coming next, right? How microbes affect human health, but specifically how they affect our neurobiology. Okay, I'm gonna get rid of that. Okay. So, you know, there's plenty of evidence. There's plenty of evidence that, you know, our microbiome influences our health in general, right? But there's an article that was published in Science magazine that I've had on my list of interesting articles for months at this point, that proposes, you know, a term that I find to be at least fun, if not, you know, a little gimmicky, but useful: the psychobiome. Okay, this isn't a term that's like widely used in biomedical science, just to be clear, but I think it does convey like a fairly widely accepted proposition, right? That the state of a person's microbiome can influence the state of a person's nervous system, central nervous system, and then therefore a person's psychological state. And so, you know, the main article I wanted to discuss isn't publicly available, so you can check that one out, the Science Mag one. So this is like pretty much the biggest chunk of the science that I want to talk about. And it's behind like a paywall, which is kind of obnoxious. And again, the science one is publicly available. It's written by a writer named Elizabeth Pennisi, I believe is how you pronounce it. And she does a good job of like touching on a lot of the rationale for why neuroscience has become interested in the microbiome at all, right? Even though it's, you know, ostensibly not in the brain. More on that later though. So, you know, for example, like scientists have acknowledged a correlation between the incidences of conditions that are like overtly gastrointestinal, right? Irritable bowel syndrome or whatever, and conditions that are clearly neurobiological, like depression. And then similarly, people who are on the autism spectrum can tend to suffer digestive conditions. People with Parkinson's disease can be predisposed to constipation. And so, you know, like a correlation between depression and the use of antibiotics, and specifically antibiotics, right? Rather than like antiviral or antifungal medications, which, you know, have no significant effect on at least the bacteria that inhabit our gut. That connection has been observed as well. And so back in 2017, believe it or not, I talked about a study that showed similar distributions of microbes among people who were diagnosed with post-traumatic stress disorder, PTSD, and people who were exposed to trauma but didn't end up developing PTSD. So, you know, no big difference in the experiences there. I believe this was in South Africa, if memory serves. But what they did find were significant reductions in 3 specific types of microbes among people who did develop PTSD, right? After you know, a shared traumatic experience or also experiencing a traumatic experience. And of course, you know, I have a link to this study in the description below from 2017 again. And of course, I also have links to all the other articles and studies that I'm describing in the discussion below. But the point being that this group studying PTSD, they identified, you know, when assessing the microbiomes of people who were all exposed to traumatic experiences, the people who ended up developing PTSD seemed to all have reductions in the same 3 types of microbes. So, you know, might it be, you know, what might it be about the presence or the absence of certain microbes that might influence our biology, our psychobiology? Well, so an article that was published in the journal Stroke argues that rather than the presence of one specific microbe, you know, being the culprit for disease states, like it's not just that the microbe is there, rather it's the presence of waste byproducts from a bunch of different types of gut microbes. Trimethylamine N oxide, TMAO, don't worry about that word, but it was that molecule that multiple microbes are able to produce, or inevitably produce. That molecule may have caused increased risk of both cardiovascular disease and stroke. And so as a result, you know, rather than looking for like a specific microbe, the specific culprit, any microbe that can produce TMAO may be associated with an increased risk of these disease states that like aren't intuitively associated with what's going on in the gut, right? Okay, so back to the psychobiome. So how is it that microbes that colonize the gut might influence the brain? So that's one way, right? Just releasing these molecules. But there's a couple of possible ways. So one is that the microbes, by being exposed to nutrients and substances that are regularly introduced to the gut via obviously diet, but also environment and the environment in which somebody lives, they take in some of those substances and inevitably convert them into new molecules. Some of those new molecules can travel through the circulatory system, through the blood, and potentially gain entry to the brain, influencing the activity of a bunch of neurons. Alternatively, right, so that's like the TMAO model. So an alternative model is that some of the molecules that the microbes produce may not even have to gain entry to the brain to influence its activity, right, just by stimulating various components of the peripheral nervous system. For example, influencing the activities of components of the vagus nerves. And by the way, the vagus nerve is one of those things in neuroscience that it seems like everybody knows. But I think it's confusing. I use the term vagus nerves deliberately. Most people say the vagus nerve before diving into neuroscience, and a lot of neuroscientists use that term too. And I think the popular conception of the vagus nerve is misguided because of that use of the term, that singular term, nerve. The vagus nerve really refers to like a large group of fibers that influences the activities of a whole bunch of different parts of the body. Like it's not like a specific spot, right? Anyways, so vagus nerves. And the vagus nerve will come up again towards the end here. But microbes within the gut may be capable of secreting molecules that somehow influence the activity occurring in various components of the vagus nerves. Okay, so another mechanism number 3, might be an inflammatory immune response to the presence of specific microbes, right? They don't cause like a full-blown illness, right, where you feel like you have the flu or you get nauseous and can't keep food down. They're just kind of like, there's like a simmering immune reaction. And evidence has accumulated that shows a pretty firm relationship between chronic inflammation and changes in psychological states like depression and things like that. So Elizabeth Pennisi, right, in her article, the top one, in Science Magazine, discussed, in her article, she discusses a company that's interested in trying to approach the microbiome from a treatment perspective, and they're not the only one. But it's an interesting company, and it's shown some impressive progress, amassing one of the largest collections of human gut microbes. But so you can check that out. I just don't think it's worth focusing on that company in particular. But one of the group's findings does strike me as rather interesting and worthy of attention. So just like the correlations of specific populations of microbes and the occurrence of PTSD, the group found that individuals with a lower abundance of a specific group of bacteria, Bacteroides, correlated with depression. So they got more granular, finding that a component of a brain network called the default mode network had detectable changes when measured via fMRI, functional magnetic resonance imaging. It's just a technology that enables us to visualize changes in essentially metabolism, changes in blood flow, basically, that we think strongly correlates with changes in brain activity. So the default mode network, this is a depiction of it. It is a whole complicated topic, but just from this picture, you can tell that it's like a pretty broad network within the brain, right? And so this is a DTI, diffuse tensor image. It's really just showing some of the main hubs within the DMN, or the default mode network, which you can see as like those yellow blobs, right there. And all those fibers, between these yellow blobs are the fibers connecting them, and sending information to and from different structures within the network. And I'm pretty sure that they're showing us the inferior parietal lobule areas, and the angular gyrus, right? Those structures, or that structure, the medial prefrontal cortex, and then maybe some early visual processing areas within the visual cortex. I'm not 100% sure. There's like no legend that accompanies this, frustratingly. Here, we can take that down, and that's it. Not 100% sure, but it doesn't matter. Like the only reason I bring up this picture is to illustrate just how distributed the default mode network is at least believed to be. And I mean, it's complicated. When I say it's complicated, I mean that it's still kind of sorta controversial among neuroscientists as to like the validity of us thinking about this giant network as like a discrete thing. But I think it's safe to say that like the broadly accepted concept of the default mode network is that it's a group of various brain regions that directly interact with one another, and it becomes particularly active when you're sort of like zoning out, daydreaming. You're not focused on any particular task or like focus of attention. And so this group found meaningful differences in a component of this network, the dorsolateral prefrontal cortex, or DLPFC. So connectivity with the connectivity of that structure with the left anterior medial frontal cortex. Okay, so these are like just two slightly different structures that aren't super far away from each other, but they're different. Specifically within the left hemisphere, so another instance of lateralization, which is becoming sort of a bit of a theme. So that connectivity seemed to correlate with differences in the presence of that particular microbe, Bacteroides. And of course, I've linked the paper below. But for our purposes, like the specific anatomy isn't like really particularly important. Rather, it's just one more correlation between consistent differences in gut microbes in psychiatric conditions and neurophysiological activity. And in this case, that those correlations of microbes in psychiatric conditions was also correlated with consistent differences in activity in specific parts of the brain, right? So cool finding. And there are other studies that have demonstrated correlations between like the presence of specific microbes in psychiatric conditions. The field is still amassing data that demonstrate the like mechanisms by which like a specific microbe induces a particular change within the central nervous system. And it is a very, very challenging effort, just given how indirect these relationships likely are. It's not like that microbe, Bacteroides, is like literally in, the DLPFC, the dorsolateral prefrontal cortex, like tweaking around with its activity, right? It's got to be some kind of indirect mechanism. And so it's really, really, challenging. But fundamentally, I think it's like absolutely reasonable to assume that like just like the food you eat can influence your health, as well as the microbes that inhabit your gut, the microbes themselves can influence more than just your peripheral, non-brain related, physiology and health. And, since the brain and body are inextricably linked, at least for now, at least for me, it's not a giant stretch to suggest that these, that the microbes that have been, cultivated and grown within a person's gut can influence brain states such that, they can become predisposed to psychiatric conditions or become resilient to them. So that's the whole concept of like the psychobiome. And that's really just what I wanted to highlight. But while I was revisiting this topic, I came across a pretty fun project that's going on at the University of Colorado in Boulder, that's exploring whether there's a microbiome that's specific to the brain. Okay, and like honestly, I have no idea if this is like still an active project at UC Boulder, and it's honestly not the kind of thing that I would devote my PhD to doing. But it's absolutely, it's like an interesting question that's like worthy of an answer. Are there microbes that inhabit the brain? Or is there like an ecosystem of microbes that inhabit the brain, akin to the gut microbiome or the skin or the mouth or the ear, But beyond that, I also think it's worth noting that, while there are like a few companies out there that have products that like might claim to have some kind of salutary, it's like physiological or neurological benefits, right, or effects, there really isn't like nearly enough evidence to be confident that anything like that will work at this stage. And like, okay, I essentially exclusively talked about human research so far, but a lot of the evidence that forms the like foundation for this field is based in animal research. And these kinds of studies can be extremely informative, but there's a reason that treatments and medications are always tested in humans before they're recommended to people, right? These what we call preclinical studies, they serve as like roadmaps, right? While they can help to guide the work of biomedical scientists, they aren't like the destination, right? But just because I happen to have met and almost worked with one of these scientists that I'm about to mention right now, let's talk about one example of this animal research. So, working with mice, the group in Mauro Costamatioli, I believe is how you pronounce it. He was known as Mauro. So Mauro Costamatioli, he's at Baylor College of Medicine, my former graduate institution. His lab found that when mice were born to mother mice, right, who had been given a high-fat diet, the offspring would consistently exhibit deficits in social interaction that sort of resemble some of the types of differences that we see in people on the autism spectrum. But the reason this study got a fair bit of attention beyond just that recognition, beyond it just being like a potential dietary connection, and the reason I'm bringing up in this context of the psychobiome, is that when the scientists looked at changes in the microbiomes of these mice, they found reduced levels of a specific bacterial species, L. reuteri, or reuteri. I don't know how you pronounce that. But, whatever. It's just this specific bacterial species. So, okay, so we have lower levels of this bacterium, or these bacteria, in the microbiome that's associated with consistent social deficits and exposure to a specific diet, during development. So the next question became, what happens if we restore levels of that specific bacterium in these mice? Lo and behold, when they did so, they saw a reversal in those social deficits, right? So, like any competent group of neuroscientists, they then asked how. It might be that the restoration of this particular bacterial species might influence something as intuitively unrelated as social behavior. And so they found that L. reuteri, the bacterium that, that they restored, influenced activity in the vagus nerves. And if they prevented that interaction with the interaction of the bacterial species with the vagus nerve, they prevented that interaction to reach back up into the brain, to influence the brain, they no longer saw restoration of social behavior. And so they argued that this is via an oxytocin-mediated effect. And so, when you think of social behavior in a neuroscience lab, you're essentially legally mandated at this point to at least mention oxytocin. But so it's a signaling molecule that's been associated with social interaction, trust, infant attachment, all kinds of really, really basic human social interaction. So they asked if mice lacked oxytocin receptors in specific parts of the brain that happen to be associated with reward. They were sort of an addiction lab before they got in, not an addiction lab, but they did a lot of addiction research before they got into this. So they know a lot about reward centers, reward circuits. And so they asked if mice lacked oxytocin receptors specifically in those brain regions, and they, again, restored levels of that bacteria in the gut microbiome, would the mice still exhibit that restored social behavior that they saw initially? Guess what they found? Those mice without oxytocin receptors in those specific reward-associated parts of the brain were no longer capable of exhibiting that recovery of social behavior. So pretty cool. That's pretty close to a mechanistic demonstration. It's not exactly a mechanistic demonstration, but it's pretty close. It's pretty good. And so that's just I remember when Mauro he actually came to Penn after I'd moved here, and he gave a talk. And I remember when he was his lab was beginning to get into this work, everybody was like, "What are you doing? You're not even a microbiologist or anything like that. You don't even sequence things." It was just sort of out of left field, but he did awesome research. It's really, really interesting. And that was just one paper. They've done several others now. So that's just one example, right, of the kind of studies that's found an influence of, the landscape of the gut microbiome over the activity in the nervous system and the brain in particular, such that behavior can be affected. But so, all of this said, fairly convincing stuff, right? Despite all of this accumulating evidence, we're still nowhere near the point of basically being able to prescribe an optimal gut microbiome to achieve an optimal psychobiome. We're just not there yet, right? We're pretty confident that such a thing may exist, and it very likely varies between individuals, but we're just not at the point where we can enumerate exactly which species would compose your optimal microbial landscape, right? And so that's why whenever you see a product that purports to seed your microbiome with healthy microbes or probiotics, you should be skeptical. They may very well be absolutely correct, right? But that's not the point. They're basing their claims on a level of evidence that we wouldn't accept for, basically any kind of medical treatment. And given what we know about the diversity in what are likely, ideal or acceptable or, optimal, whatever you want to call it, populations of microbes, given that there's so much variability in those, I think it's safe to say that it's unlikely that there's going to be a perfect one-size-fits-all product on the market that'll work for us. And I'm happy to talk about what I could imagine that market beginning to look like. Because it's interesting, and I think it's totally viable and totally doable just with the technology we have today. We just need more knowledge regarding how to identify optimal microbes given a person's unique biology and neurobiology. But topic for another day, I suppose. So that's what I have for you. If you stuck with me and you haven't subscribed, maybe you'll consider, prodding the subscription button or probing it, poking it, punching it. You get the idea if you haven't yet. Otherwise, let's start the live half. Okay. Hello, everybody. My name is Ian Mclaughlin, and I have a PhD in neuroscience from the University of Pennsylvania here in sunny, beautiful Philadelphia. And on YouTube, youtube.com/anthropoid, I just talked about the psychobiome. So this is a term that refers to the relationship between the gut microbiome, or just the microbiome in general. It doesn't have to be the gut microbiome, but the microbiome and the activity of your nervous system, in particularly the central nervous system, particularly the brain. So where am I going to go after Periscope ends? I thank you for bringing that up. Well, in part, I'm on YouTube right now. So, I just talked about the psychobiome. It's sort of a made-up term, but hey, a lot of terms in science are made up. So youtube.com/anthropoid, you can find that via my profile on Periscope. But then also, I've been working with, hey, what's up, Scott? Good to see you. I've been working with a totally new platform called Happs, HAPPS.TV. It is fundamentally a live streaming platform, but it has a lot of tools that actually Periscope does not have. Pretty small community, and I really like their sort of philosophy and their mission. And it's a cool community. I mean, a lot of people from Periscope have already migrated over there. If you like science-related stuff, Isa Betabug or Isa Betancourt is over there. We're trying to convince, or we have successfully convinced DC Hooper. I should say, Isa Betancourt is an entomologist. Used to live in Philly. We collaborated a few times. Now she's in Michigan. But then Deanna Hooper, a cosmologist who studies the universe, dark matter, dark energy, these types of things. She's going to be joining us, or trying to cultivate a little science cluster. So you can get to Happs via also my profile. I have two links on my profile. One is to YouTube, which again, youtube.com/anthropoid. The other one is Happs, HAPPS.TV. Whoopsies. I can hear that my daughter has awakened. The Kraken has awakened. So yeah, I thought Parler was where all the tools were. That was a pretty good one, Scott. That I think is better than most Parler jokes I've seen. So there were a couple other really cool things. So what are y'all gonna do when Periscope ends? So that's what I'm talking about, Brittany. Happs.tv or youtube.com/anthropoid. That's where I have specific topics. I talk about them sort of at length. Today I talked about the psychobiome. And so you can click on or tap that, click it, however you interact with your devices on my profile page in Periscope. But then also Happs.tv. Okay, so there was a really cool study that came out that was sort of surprising. That basically showed, it wasn't a study, it was like a case study, okay? And basically there was a case of a guy who, youtube.com/anthropoid. So you can just go to my profile page on this, on Periscope, and you'll see the YouTube link, just tap that. Guy had leukemia, okay? And it was pretty advanced. And they showed like CT scans, X-rays or CT scans, I can't remember, probably CT scans, of the extent of his leukemia, and it was pretty bad. I think he was in his 60s, or pretty extensive. Guy catches COVID-19. Okay, not good, right? Not optimal state of affairs, and his leukemia went into remission. What? So crazy. I'll share that on Twitter because it is kind of baffling. And so it's just a case report. They don't really have a mechanism. They don't understand why this would even happen. But their assumption is exactly what I would think would be the assumption. It's that COVID-19 induces this radical change in the immune system. And in this very narrow specific case of this very specific person with this very specific leukemia, their leukemia was attacked by this elevated immune system related activity. Really cool. Certainly not the kind of thing that you want to go out and get COVID-19 over. But just like has been the case now for months we're learning a lot about human biology, the human body by understanding COVID-19. It's just such a weird virus. Would you take the vaccine? Yes, I would. I absolutely would. By the way, so I've been talking about the COVID-19 vaccines in the United States, focusing on the mRNA-based vaccine. So that is the Pfizer vaccine and the Moderna vaccine. Those are both mRNA-based. They're rather similar. There's obviously some differences, but they're rather similar. But we've been hearing about this Johnson & Johnson vaccine. Well, it turns out that the Johnson & Johnson vaccine is also an adenovirus-based vaccine, just like the Oxford AstraZeneca vaccine. Pretty cool. But what's extra cool about the J&J vaccine is that it's a one-shot job, right? You just get one shot and you're vaccinated. So we haven't seen the reports of the clinical data. I imagine we're probably going to be seeing that pretty soon, though. Also, the J&J vaccine does not have to be stored at like negative a trillion degrees. It can be stored in a refrigerator for months. And so that would be like a complete game changer internationally. So, in other words, it's kind of similar in methodology to the Oxford AstraZeneca, the one that's being produced in the UK. But it seems to be more sort of resilient and therefore the logistics will be significantly, less challenging. You won't have to like keep it like, in like liquid nitrogen, basically, otherwise risking its deterioration. Yeah, where is Abyss? Hey, Saucy Mona Lisa, good to see you. I'm not sure. I mean, people become busy these days, which is understandable. Okay. So glad to see you on this fine Sunday. It is a fine Sunday, isn't it? Thank you, Amanda. Likewise. What is the best treatment now that I caught it? 60 Healthy Runner. Power Realty OC. Well, of course, the way to move forward is to engage, I presume that you've at least been tested, right? So you're certain that it is COVID-19. In which case, call your primary care physician. If you don't have a primary care physician, go to your local, cities or your counties or your state's health department for guidance on like what to do. Because obviously we're still seeing, alarming rates of it spreading. And it really just depends on your symptom severity. But obviously the optimal thing is to go work with or consult with a physician. But broadly speaking, it's if symptoms aren't severe, what's going to happen is it's just like recovering from any other virus, right? Because there are some treatments, but they're really for severe cases. And so if I were talking to a member of my family, that's what I would tell them. See what your local guidance is. The reason that you want to check your city's Department of Public Health or your states or your counties or whatever is that they'll be more aware than your average bear of the sort of status of the hospitals in your area. Like, are they packed to the gills or are they not? Like, there was a long period in Philadelphia where while other large East Coast cities were getting like just hammered and the hospitals were saturated, the ones in Philly were not. And so the Department of Public Health had to come out and say like, hey, like I know everybody's really concerned about filling the hospitals and you don't want to overload the system, but we're not like that. Like, definitely if you get sick, go to the hospital. So the point being, like, the guidance will vary depending upon where you are in the country. Now, apart from that, I mean, again, like let's say I were to catch it, right? Knock on wood, this is not a virus you want to catch. Here's what I would do, right? And my symptoms weren't particularly severe, right? You're in a different age category. You're not quite in the like overtly high risk category, but these numbers are, the boundaries aren't like very firm, right? But my symptoms weren't severe. I would do exactly what I said, contact my primary care physician, and keep them up to date. Obviously, keep doing temperature checks, these types of things. But primarily would be make sure I'm eating a lot and sleeping a lot. That is like the best thing you can do, when you have pretty much for your immune system in general, right? And when I say eat a lot, like eat a lot of competent, a competent diet, not just like a lot of bagel bites, right? there's a kind of like a reason that chicken noodle soup is like the go-to, at least in the United States, for like your sick food dish, because it has like a pretty good distribution of fats, proteins, obviously, carbohydrates. It has some electrolytes, right? The salt and stuff like that. Some micronutrients and, whatever, the carrots, the celery. That's what you want. You want, like, not necessarily chicken noodle soup always, but, like, a good distribution of nutrients. If you're a runner, I assume you probably do pay at least a bit more attention than the normal person, your typical person who doesn't exercise like that or maybe compete like that does. That's what I would do. But, it is something, you just want to monitor every day. Okay. What are your thoughts on Xanax? Hey, Sasimon, Lisa. Yeah. What are your thoughts on Xanax? So, I have thoughts. So Xanax, just for everybody that's unfamiliar, Xanax is alprazolam. It's in a part of the family of medications, of drugs called benzodiazepines, which includes equally infamous substances like diazepam, which is Valium, or clonazepam, which is Klonopin. You might have now recognized a sort of theme to the naming scheme, right? Azepam, that's generally speaking a benzodiazepine. So alprazolam is Xanax. Alprazolam is... why do we have so many benzodiazepines? They all essentially bind the same part of the GABA receptor. It's called the benzodiazepine binding site, or the benzodiazepine site. Weirdly, right? It does not bind where GABA binds. So GABA receptors, GABA is the nervous system's principal inhibitory neurotransmitter. So when GABA is being released, neurons begin stop firing so many action potentials. They're not firing as many impulses as you might call them. And so why do we, what's the difference between like alprazolam and Xanax or diazepam and Valium? So Valium is very long lasting, and it has a very interesting reason for that. I mean, it's interesting to nerds like me, but basically it's because diazepam, you eat it, right? It itself has a pretty long duration of effect, but then our body converts diazepam into another benzodiazepine. I can't remember if it's oxazepam or lorazepam, but it's one of the two. And then that, not all of it, but a lot of the diazepam becomes another benzodiazepine, and then that, therefore, has its own pharmacological effects. And so it's sort of like taking two benzodiazepines. Alprazolam is a shorter-acting benzodiazepine. About 6 hours, I believe, is the half-life. But fundamentally, they do the same thing, right? I mean, it's just, apart from that difference in duration of effects. Okay, these are useful medications. These are medications that used to be dramatically overprescribed. I believe in the past they were called Mommy's Little Helper, because they were just prescribed to like every housewife in the country. Well, every housewife in the country who had access to that kind of, physicians who were able to prescribe drugs like that. And so as a result, we learned a lot about the development of substance dependence when it comes to benzodiazepines, which is unfortunately rather severe. So benzodiazepines, because of the receptor they bind, just like you develop a tolerance or substance dependence and potentially addiction to alcohol, you develop a substance dependence on on benzodiazepines, including alprazolam. And, that's true for essentially every single drug that people take for fun, or that, that is psychoactive, that influences the brain. But withdrawal from some substances can be life-threatening. Withdrawal from other substances that isn't. There is a cannabis withdrawal syndrome, but it's not nearly as severe as benzodiazepines or opioids like heroin or, cocaine or alcohol, certainly alcohol. But there is a withdrawal syndrome. When it comes to, nicotine withdrawal is not fatal, or not potentially fatal, but it is super severe, right? When it comes to alprazolam, Xanax withdrawal, and alcohol withdrawal, by the way, these are sort of in a unique category of withdrawal syndromes that can be life-threatening. And here's why. As somebody's been exposed to these substances, this is true again for Valium, for Klonopin, for Xanax, and alcohol. As people have been exposed to these substances for a long period of time, generally high doses of these substances for a long period of time, their brain becomes accustomed to the chronic presence of that substance, and because the thing with which these substances interact primarily is the GABA receptor, the brain adapts and modifies the sort of GABA landscape. It modifies the number of GABA receptors that are available to be bound by drug and neurotransmitter, GABA itself. It also, the sensitivity of the GABA receptor can be altered, right? Basically so that the brain is adapting to reapproach what we call homeostasis, to behave or to exist as though it existed before, somebody had ever been exposed to the drug in the first place. And so the problem is that then when you take, so that's fine, right? And so long as, somebody has sufficient access to these substances, it's not really, it can be a problem for other reasons, but it's not like a life-threatening situation. The problem happens when the substance is withdrawn from the equation and the brain is no longer getting exposed to this benzodiazepine. The problem emerges because all of those adaptations, all of those changes are still in place, and they take a long time to get there, and they take a long time to go away. And that's why you have a withdrawal syndrome. And when it comes to the withdrawal from, and that's the fundamental mechanism for all substances. But the problem with benzodiazepines and alcohol is that because it's a GABA receptor, you can risk having seizures and potentially die. So that's, sort of the central problem with benzodiazepines. That's why physicians don't prescribe them nearly as much as they used to. But I think it's also worth acknowledging, I'd be remiss if I didn't, that, we're, I think, all becoming more and more familiar with the opioid overdose epidemic, opioid addiction epidemic, but also opioid overdose fatality epidemic. Benzodiazepine addiction has also been increasing throughout the United States. Not at the same rate and not at the same severity as opioid addiction, but it is expanding. And in fact, the UN, who monitors, like the availabilities of various medications, not just substances that people use recreationally, but medications in general, they have detected, they have noticed that there's like a pretty substantial decrease in the availability of benzodiazepines in particular. A lot of interesting potential explanations for that, but fundamentally it probably means that more people are taking benzodiazepines. Excuse me. So yeah, I could go on, about why they're useful drugs, why they're not fundamentally evil and stuff like that. But that's the sort of landscape of benzodiazepines. I believe somebody was calling in. I'm sorry, I didn't grab that. I think I caught it right as the number disappeared. So do feel free to call in again, and I will immediately respond. Okay. My friend was addicted to benzos and opiates. He killed himself about 2 weeks ago by overdose. Oh man. Sorry to hear that. Yeah. That is one of the riskiest combinations. There's like maybe that is the riskiest combination. Pretty much every time you hear of a celebrity overdose fatality, without exception, almost, it is going to be associated with a combination of benzodiazepines and opioids. I think that was true for Heath Ledger. I think that was true for Philip Seymour Hoffman. And sometimes also it's because they didn't know that there was fentanyl in the opioids that they were taking. Okay, sorry about that. Have any tips or tricks for someone starting in a neuroscience research lab? Whoa, Grace, congratulations. That's awesome. I have thoughts. And then we could talk about what benzos are used for. No problem. So, here's my tip. First, depending on if you're a grad student or a tech or something like that, first is do not be afraid to ask questions. Don't, I think a lot of us who come up through the educational system in the United States are like, we have like a phobia of revealing that we don't know the answer to a question. I definitely did. That was definitely me. But the problem is that you can end up wasting weeks and weeks and weeks on a problem that could have been solved within a 10-minute conversation, right? So that would be number one. Number two is be open to collaboration with people in your lab group, but then also outside of the lab. I did that all the time, and it was so beneficial. It's beneficial not only for my specific project, but also it made me a better scientist. I got exposed to so many more methods than I would have been exposed to if I were just like in my own little silo, which is, I think, the norm for a lot of neuroscience researchers, is that there's just this we're scientists. We like to sit in our tower and conduct experiments, right? But so collaboration is key, and read a lot. I think most neuroscientists, that's not a surprise, and it's just sort of necessary for success. But read reviews, and to try and get a sense of even tangentially related domains of research. Like, let's say you're researching addiction, right? Like I did. I was also interested in how these brain regions that regulate emotion and obviously addiction and reward and stuff like that, how they also influence movement, like motor behavior. And so I read about that. And then my project ended up veering towards this motor response in an addiction, or in an anxiety, assay, behavioral assay, and it ended up being extraordinarily valuable. And then my viral tracing ended up revealing that a lot of those brain regions involved in motor behavior were connected to my circuit of interest. Nobody had really found that before, and so those would be like my main ones. But the main one is just don't be afraid to ask questions. Don't try and solve things on your own, of course, but most people, at least in my experience, yes, Michelle, please do feel free to call back in. I'm sorry I missed you the first time. You try and solve things on your own, of course, but most people in the lab are like super open to collaborating, right? It can be a pretty solitary experience. My friend's wife is on the wait list to start psilocybin therapy. What? That's amazing. Very interesting. I hope you'll let me know how it goes. Like to contribute about benzos? Yes, please, Michelle. I'm sorry I missed you if you're still here. Did he say nicotine addictions can also be fatal? I don't know if I heard that right. No. The opposite. Nicotine withdrawal is not fatal. It's very, very severe. It can feel fatal, but it's not. Even withdrawal from opioids isn't life-threatening in the way that benzodiazepine withdrawal and alcohol withdrawal is. Okay. So that fame is modern tragedy. Yeah. How badly the human brain is affected by depression if lived for 85 to 90% of the lifetime so far? It's an interesting topic. It depends on how, 85% of my daughter's life is not particularly long. She's 2 years old, so I don't think she's very fluffy-tailed at the moment, knock on wood. But as opposed to somebody who's had depression for, 40 years or something like that, it has, detectable, or it correlates with detectable changes in the brain. The one that I think is most intelligible to most people is a change in synaptogenesis within the hippocampus. Like that has just been detected, and like hippocampal volumes have been shown to be decreased in in the brains of people who have severe depression. And then of course depression can be associated with all kinds of changes in diet. Very importantly, it's one of the classic symptoms of depression is perturbed sleep patterns, and it can sort of go both ways. The sort of classic one is people that just tend to like wake up after like 4 hours of sleep and they just cannot get enough sleep. That can be a symptom of depression. But it can also be the opposite, hypersomnia, where like people just want to sleep for like 15 hours, and just like do not want to get out of bed ever. They just don't have energy. So obviously that has all the indirect ramifications of insufficient sleep. Okay. I find him interesting. Oh, wait. Okay, let's see. I find him interesting. So what is your interest in addiction? I guess I'm not sure, because I thought this stuff would have been discovered in the MK Ultra program. So MK Ultra is a very interesting topic. I'm not sure what you mean by him. If you're bringing up like specifically like a specific person. But for anybody that's not familiar, MK Ultra was an entire project by thank you. Yeah. Well, you thought most of this stuff would have been discovered in the MK Ultra program. Well, the MK Ultra program was basically, there weren't a lot of scientists involved. Like there were, but they weren't neuroscientists. They weren't like actual neuroscientists. They were mostly chemists and like the intelligence community. The goal was to develop some kind of mind control platform. It's an interesting story. There's a great book that I always talk up called Poisoner in Chief by Stephen Kinzer. I think Kinzer or Kinsler, who's a professor of, I believe, journalism or history at Brown University. He's a former journalist. Great book about, I think his name is Sidney Gottlieb. Sidney Gottlieb was the chemist of the CIA. He was like the head chemist of the CIA. He was responsible for synthesizing a lot of poisons that were used by the CIA or have been used. But then he was also responsible for, ushering this project in pursuit of a mind control platform, because at the time the United States was or the government at least was convinced that the Soviet Union had developed some kind of mind control. And so they were like, oh, we got to do something. And then, of course, happening at the time was the sort of the nascent expansion of the availability of psychedelic drugs. Sidney Gottlieb, working for the CIA, was basically the reason that like a lot of people during that hippie era were exposed to LSD. The CIA purchased all of the LSD from, I guess it was Sandoz Pharmaceutical, for something like $100,000, and it was a ton of LSD. But anyway, so a lot of the beat poets were exposed to LSD via like these tests from the CIA. Obviously, it was a miserable failure because there is no such thing as a mind control device or platform, and certainly if it existed, it would not be LSD or psilocybin. So MK Ultra is alarming, but there were even funnier and more alarming projects that happened. Yeah, Tim Leary, of course, was a major figure in this. My favorite was Operation Midnight Climax. If you've never been aware of Midnight Climax, basically the CIA was running a bordello in San Francisco, and they had set up basically like one-way wind mirrors. What are they called? One-way, where you can see only through one direction of the mirror. And there's this story, and so patrons would come to the bordello and patronize it, and so there would be people from the CIA. There's like a story where there's like a guy in the CIA holding a martini, just watching they would dose the patrons. They would give them LSD, and then they would just watch what would happen through that window, just drinking a martini. It's just like a perfect story to encapsulate that era in politics. But yeah, that was a good one. Operation Artichoke was a good one. There's a bunch, but the point is, almost nothing was learned from those projects. They were miserable failures. What was learned is that like LSD makes people giggle a lot, and like they became, oh my god, I think I talked about this on YouTube or on a podcast. I have a podcast called Wired to Be Weird, but I'm pretty sure it was on YouTube. Yes, it was on YouTube, youtube.com/anthropoid. Well, whatever. I don't know. There's a passage of like their findings from all of these projects of like, okay, so are we going to be able to use this? The Dulles team is like, are we actually going to be able to use this? And they mandated it. They mandated that they not be told everything because they'll what mama don't know, right? So they wouldn't be culpable. And like the sort of like executive summary, right? The sort of summary of their learnings was that like it rendered people rather unreliable, which is just like, yeah, because they were like tripping, like losing their mind. Anyway, super funny era, if not a little scary. Makes you wonder what might be happening now, but at least there's no evidence. But yeah, they weren't really like studying it. And tragically, because at least in part because of that and the church committee hearings and just the sort of reaction to the hippie generation and the excesses of the generation and the tight association of LSD and psilocybin and all these substances, mescaline, with that movement, there was this like, it was taboo. It was like verboten to study those substances. So we went basically several decades where it was, and of course they became as illegal as the substances can be, right? So we went decades without actually studying these substances because nobody wanted to spend their career doing that. That's changed now, though. So now there's a lot more research going on regarding psilocybin. I think it's safe to say more so psilocybin than LSD, but still both. And then also other molecules that would be in the same category of substances. And particularly, as we heard, Sassi Mona Lisa's friend is going to be engaging in a clinical trial with psilocybin for the treatment of depression, which is excellent. A lot of really, really strong data coming out of those clinical trials for both psilocybin, which, if you're not familiar, is the sort of active ingredient of magic mushrooms, but then also MDMA, which is sort of in the same category. It's not quite a psychedelic, but it's sort of in the same category of substances. Different pharmacology, blah, blah, blah. But He's rich enough to have that background, so yes, it's real. No, I definitely would not call myself rich. You don't go into academic science to become rich. No, I live in a very small apartment on sort of the outskirts of the city of Philadelphia. Center City is what we call it, Central Nugget. And, we're a dual income house, and we like it here. So, okay, can stress and overthinking cause lack of sleep? Absolutely, no question, definitely. Chronic stress. So like, when you're talking about chronic stress, generally speaking, hey, it's good to see you. Actually came up earlier. Good to see you, man. I was just talking about the Poisoner in Chief, that book about Sidney Gottlieb and like MK Ultra and stuff like that. So you missed a pretty laborious, tedious explanation. But anyways, so like chronic stress does a lot of things. There's a lot of biology that or that is changed by chronic stress, but probably the central one, which is sort of like chronic stress can beget stress, is the glucocorticoid signaling system. So basically, what happens when you experience an environmental challenge, right? That's what stress is that your brain and the adrenal glands outside of the brain, but whatever, this like extended network of neurons releases a bunch of neurotransmitters and hormones, right? It releases norepinephrine, it'll release some dopamine, some epinephrine in the brain, but then it'll also induce the release of cortisol. And so cortisol is a hormone that will circulate throughout the body and influence a bunch of different things, ultimately to prepare you for battle, right? Like, these are systems that evolved hundreds of thousands of years ago, right, to prepare our primitive primate relatives, ancestors, for survival situations, fight or flight. But so the way the system works is that so you've had this surge of glucocorticoids, surge of cortisol, and the evolutionary point being that you are prepared for an acute challenge to your survival, right? That you then take some kind of action to extricate yourself from, right? To avoid, to either run or kill, right? But then that stress is supposed to, that environmental challenge is supposed to abate, right? Once it's no longer there, once you've solved the problem in one way or another, there's no longer any need for that elevated cortisol signaling. And that's important because part of what those molecules do is they like inhibit digestion. They inhibit nutrient absorption. They inhibit the immune system. They change the way blood is distributed throughout the body. So they alter metabolism, and that's all fine, well and good when you're like in the moment, the survival moment. But afterwards, you want to be able to digest properly. You want to be able to relax. You want to be able to absorb nutrients, and parts of the body that were recently deprived of normal amounts of blood need to get their blood back, right? And so what happens is that a part of the brain will detect the elevated cortisol, and over time, that process of it detecting that cortisol, it'll begin to inhibit the process. So it's called negative feedback loop, right? You get an increase in one neurotransmitter that then causes an increase in activity here, that then causes a reduction in activity of that neurotransmitter, right? In this case, we're talking specifically about cortisol. So what happens when you're chronically stressed, right? Where that environmental challenge, perceived or otherwise, remains, right? You continue experiencing abuse from somebody in your family or in school or whatever, or you continue regularly don't know when you're going to get your next meal. These types of things. You're living at a level of precarity, of financial precarity, where you just don't know if you're going to be able to pay the rent, to provide for your family next week, right? Those are real challenges that will activate that stress response. But if they don't go away, what happens is that mechanism that detects elevated levels of cortisol, increases activity, and then reduces levels of cortisol, that sort of atrophies, essentially. It sort of becomes less capable of doing that. And then therefore you become less capable of reducing that stress response. And so you're just perpetually stressed, even though you no longer actually rationally, consciously experience that stress. You have plenty of, you're financially secure, you have plenty of food, you are no longer around somebody who has traumatized you. So that is sort of like the kind of the inertia of chronic stress. And so therefore all of those things that I was talking about, reduced nutrient absorption, altered metabolism, altered digestion, inhibited the immunosuppression, inhibited immune system, all that stuff just stays at that state, and that can be rather unhealthy. Okay. Not true, God makes it happen. Or, that's an explanation too. Okay, I have so many questions. Well, let's chat. It's my favorite topic. Also, I just had to accept that there's probably a camera behind the mirror whenever I go to a bordello. Yeah. Can you explain to me original thoughts? How do they come to be? I tie that to spirituality. Well, okay. I mean, if you already have a formed opinion about it, then why are you even asking me, right? So here's what I would submit to you. That spirituality aside, I'm not an expert in that. I would never be so arrogant as to suggest to you that I know those answers and you don't. But what I do know a lot about is the brain. It's my job to know a lot about the brain. I spent basically 10 years focused on it. And what I can tell you is that there is evidence that specific processes within the brain have been associated with, original thoughts, right? Creativity. There's different forms of creativity, but I think that's what we're talking about, right? A creative thought, a thought that, surprises people because they never would have thought of it. And there's, again, different versions of creativity, and it has been associated with different patterns of brain activity, different strategies, right? So like one strategy, for example, is basically what we would call blind variation. Basically, it's just like uncontrolled, random, conceptual combinations, right? Where, it's just a process that occurs in the brain, particularly when the default mode network is active, when a specific network within the brain, I'm showing it back up on YouTube if you want to check it out, youtube.com/anthropoid. It's a network of brain regions that spans essentially the entire brain, but it doesn't include the entire brain, but certain parts of the brain that, some in the front, some in the middle, some in the back, right? That becomes extra active when you're kind of calm, right? When you're introspective, when you're daydreaming, when you're not focused on a specific task, when you're not being stressed out, when, you're not, when you're just sort of like drawn back, meditating, right? That can be associated with DMN activation, right? And when that network is active, that blind variation, that just sort of like random combinations of thoughts can tend to happen more. And so that's one of the, like I've heard creativity researchers, actually one over here at Drexel University here in Philly, calls it like the shower insight moment, right? Where you're taking a shower, like, that's not an original thought, that like I have my best ideas when I'm taking a shower, or, whatever, when I'm doing something that has nothing to do with my work. That's why, because when you're in the shower, you're just sort of like zoning out. You're not really thinking about like, specific tasks. You're not working on something, right? And so that's why the default mode network, that's one theory, is that the default mode network becomes active for a prolonged period of time, and that sort of increases the likelihood that some random combination of these thoughts, these experiences, these memories, produces something of value, right? Something, a unique, an original thought. Another one, though, and that whole process is a little bit more complicated, right? There's selective retention that we also have to execute, that involves like a certain component of the default mode network, and another part of the brain called the bilateral inferior frontal gyrus. See, we have trouble with these laborious words as well, but whatever. It doesn't matter what the brain region is, but basically it's that, it's you can think of it as that like a part of the brain is sort of monitoring this process, and all of a sudden when there's like a little bleep, a little signal of like, hey, wait, this is a meaningful combination. This is good. This is exciting, right? This is rewarding, this experience. Then that selectively retains that creative thought, or that thought that therefore is deemed creative. Okay, so that's one. But like, and so like a lot, that is sort of like a strategy for creativity that relies on what we call insight, right? But as Louis Pasteur, the famous scientist, would argue, chance favors only the prepared mind, right? That's his quote. And of course, he's one of the most important scientists that has ever lived. And of course, his point is that like, just sitting in a room, without any kind of exposure to new information, without learning new things, without having unique experience or, novel experiences with, new people, conversations, bouncing your thoughts off of the thoughts of others. Without that, then nothing interesting is going to happen, right? And so, and fundamentally, there's this tension or this dichotomy in at least some creativity literature. Research that is basically like there are two strategies for pursuing creativity. One is what I was just talking about, relying on insight, relying on just, zoning out, relaxing, taking it easy, taking a shower, whatever it might be, going for a walk, that type of thing, versus analysis, versus like actually trying to, enumerate all the components of whatever whatever thing you're trying to solve, whatever creative insight you're hoping, whatever creative event you're hoping to experience. Writing it out, being very meticulous about it, and then working to grand accent. Why? Thank you. Merci. I don't speak French, but anyway, so being very meticulous about it and solving problems in a deliberate way, and this is what I think about when I think of a company like IDEO, which is basically like a consultancy, a company that's very, very successful. That's literally the service they offer you is like, we're just really, really smart and creative people. But it's more meticulous. It's more analytical. It's not the shower insight. It is the analytical strategy. So all of this to say, creativity is not just one thing, and it's not like there's a switch, but it's not like there's only one way for a person to be creative. Rather, it is that what we deem to be creative emerges from patterns of brain activity that have been detected, that have been characterized, and at least for me, that the reason I find this literature interesting is that it influences how I try and be creative, right? I try to do a little bit of both. Importantly, there's no evidence that either strategy is superior to the other, right? Meaning that like you might think that like the insight-based strategy might be better because it just sounds like it's more creative, right? It just sounds like more like the eureka moment. And brilliant, but it's not superior to sitting down, writing out your problem, and finding the best solution, a creative solution, to a problem. Okay, I just haven't talked about that literature in a long time, so thank you for bringing it up. It's a really interesting topic. Do you think creativity could be accelerated in synaptic pruning if synaptic pruning was delayed in adulthood? I'm curious. Yeah, that's something I thought about. So what Abyss is talking about, synaptic pruning is the process by which a lot of the connections between neurons within the brain are pruned away or removed. And that happens in basically two main periods of life. It's happening in my daughter's brain. Well, it's actually not happening in my daughter's brain, but early on in life. Well, it is. It's always happening. But early on in life, there's a period where the brain undergoes synaptic pruning. And then particularly during puberty, that's where like a lot of synaptic pruning occurs. And the flip side of that is that therefore children have far more synaptic connections within their brain than adults do. They also have far more excitatory activity in the brain than adults do, which is part of the reason for why children can grow out of epilepsy, because the amount of excitatory activity declines as we get older inevitably, and therefore they might, while they're, when they're 9, 10, whatever, and they're above a threshold where, epilepsy is a condition where seizures occur, where there's like sort of unusual runaway electrical activity within the brain, where, when they're 9, 10, they're above this threshold. As the excitatory activity decreases, as the synapses are pruned away, they fall beneath that threshold and they no longer experience seizures. But that's why. So what Abyss is suggesting is that maybe, if we have, maybe those increased synaptic connections actually have some kind of advantage, and therefore, and the advantage would be greater connectivity, greater communication between different brain regions. I think there's something to it. I mean, I do. Clearly there is. I never really think about it this way, but there's likely an evolutionary explanation for why synaptic pruning occurs to the extent that it occurs. And by the way, I should note for everybody else, like that period of pubescent synaptic pruning during puberty is associated with the emergence of a lot of psychiatric conditions, particularly schizophrenia. So, but then also autism, bipolar disorder. A lot of psychiatric conditions actually only manifest after puberty or towards the end of puberty. And so the theory is that that process of synaptic pruning, where all those connections are pruned, not all of them, but a lot of them are pruned away to focus, to hone in on the minimally necessary circuits within the brain, the ones that have been used, the ones that are essential. If that process, which is genetically, which is encoded by your biology and your experience, but your biology as well, if it goes awry, then you can have a synaptic, architecture that is associated with some kind of psychiatric condition. But anyways, my speculation would be that synaptic pruning, the brain is extraordinarily expensive, calorically, and it's expensive as an adult. Like, we spend a lot of our energy just feeding it, right? And so I would imagine that with greater connectivity and everything would come, other odd, deprivation of resources to other parts of the brain, or, maybe it wouldn't. And so long as you could just like get enough nutrients, then you would be fine. I'm not sure. Pure speculation. But I would imagine there's something to that, man. Yeah, I don't know so much about the just elevated excitatory activity specifically, or glutamatergic activity, but increased connectivity, I think there's something to it. All right. Will the vaccine do anything against the COVID proteins that already passed the blood-brain barrier? It's a very interesting question. So Yang Gang is referring to a study that came out, 3 weeks ago now, that showed that cookies are good for the brain. I agree. they're not, but they're good for the soul. How about that? That a part of the SARS-CoV-2 viral particle, the S protein, the spike protein, part of it is capable of passing the blood-brain barrier. And then there was another study that came out more recently. By the way, the senior author on that paper was Akiko Iwasaki, who's become like a very famous immunologist in the era of COVID. But anyways, they showed using organoids, one of the coolest things in the world. Here, I'll see if I can find it on YouTube so that you can see it, because it is really neat. Here we go. Boom. Organoids. You can see it on YouTube if you'd like. They look weird. They look like pebbles, actually. That's what they look like pebbles in a river or something. Fleshy pebbles. But anyways, they showed that SARS-CoV-2 can infect neurons. So, that neurons that are organized in an organoid. So basically like a tiny little mini brain. It's like a 3D structure where neurons can form connections with each other in 3D space. Meaning that the virus itself can infect neurons. Potential implications peripherally, of course, but also potential implications for some of the olfactory nervous system. But then also, if the spike protein, part of the spike protein, is capable of crossing the blood-brain barrier, is it possible that the viral particles itself, in just one or just very, very few, by chance can cross the blood-brain barrier? That is not established. So will the vaccine do anything to prevent that? Theoretically, of course, yes, because it would help to prompt your immune system to react much more rapidly to prevent the rapid proliferation of SARS-CoV-2. So you just have fewer viral particles in your body, right? So that's one. Another is that, like, depending on certain kinds of antibodies, basically what they do is they just like decorate a pathogen. It's not like they're like it's not a scalpel. They're just sort of like, it's sort of like in American football when somebody gets when they're doing like a kickoff and a return, and when they tackle the guy who's returning the ball, and it's just like 50 guys just like jump on top, just prevent that dude from moving anymore. It's not like they untied his shoelace, or it's not like they one guy just surgically sort of took him out. It's like the whole team just sort of like amasses and covers it and prevents him from interacting with the sport anymore, right? That's kind of like what a lot of antibodies do. And so, theoretically they could, and given the fact that that at least some of these vaccines are designed to specifically target the spike protein, it might be that those antibodies would selectively just decorate all those spike proteins and just prevent them from interacting with whatever they interact with to get into the brain. So it's theoretically possible, but I mean, you know, the reality is the main effect of the vaccine is to reduce severe symptoms. That's what it does. That's the primary benefit. And I think it's also and also neurological symptoms tend to present mostly and I can show the SARS coronavirus particle on YouTube too. It is mostly people who have severe symptoms, severe other symptoms, right? Non-neurological symptoms that tend to also present with neurological symptoms. There are cases that I've talked about where like people would present in the hospital with exclusively neurological symptoms first, right? So they come in with confusion, they look like they have dementia or they have a stroke, and then eventually they do present with severe symptoms of COVID-19. But the point being that generally speaking, it is people that react particularly poorly to the virus that tend to exhibit neurological symptoms. But again, you know, as much as we know about this virus already, like it's only been around for it's only been around for like a year, right? Just over a year, pretty much. And so, you know, a lot of this is just correlational, and you know, we can't know for sure. But if your question is, you know, then let's say, you know, the vaccines don't protect against any neurological symptoms, is it worth it anymore? Yes. Like, yes, absolutely. Read today that some blind people can develop echolocation. How common and how easy for a blind person. Yeah, so that's really neat, isn't it? So when you say echolocation, oh, you know what it's like? It's like, what's that superhero? The Daredevil? He's the one that's blind, I believe, where they use... you know, it's basically, you're right. It's echolocation. Like it's basically they'll make a sound, a click, or you know, a snap, or they'll tap, you know, a cane on the floor or something like that, and they'll be sensitive to. They'll develop a sense of space, a spatial awareness, and that's been demonstrated pretty robustly. Like you know, there are videos of like somebody who's you know deaf riding a bike doing that, which is really remarkable. There's a lot of interesting things that can happen in the sensory nervous system. So first of all, the visual system is gigantic, right? It's like a third of our brain is devoted to vision, just vision, different parts of vision, right? But you know, basically the whole back of the brain is vision. And so when that is our dominant sensory system, right? We rely more on vision than any other system. And so as a result, when, you know, somebody who's blind, depending upon how, you know, how their blindness arose, is it damage to the eyes? Is it some kind of, you know, problem in the visual pathway from the eyes to the occipital lobe? Is it in the occipital lobe? And so on, right? Depending on that, you have a large part of the brain that's just not receiving as much activity as it normally would. And so naturally, other sensory systems can invade, can co-opt those brain regions. And so, right, because you don't have your vision distracting you. Yeah, basically. But I mean, you're right about that for sure. But like, but there's also a neurophysiological event that can occur. And so basically what we're talking about is like sensory substitution, where, you know, not only it is possible that somebody can use that method, that echolocation type method, and actually experience visual experiences, right? And so that phenomenon is so substantial, the ability for one sensory system to sort of adapt, co-opt the neurological activity of another part of the brain, another sensory system, is so robust that, you know, there are companies that are devoted to developing, what's it called? It's called BrainPort. I think it's called BrainPort. That is essentially a palette that somebody would wear on their tongue that has like a whole grid of electrodes. The tongue is extraordinarily sensitive to tactile, right, to touch essentially. right? There, different parts of our body have different levels of sensitivity. And when I say sensitivity, I mean the ability to discriminate, a point here on that part of the body being poked and a point like right here, like a micron away, 100 microns away, an inch away, and so on. Depending upon how sensitive that part of the body is, that part of the body can discriminate really, really, really adjacent points. And so, other part like our lips are also very sensitive. Our hands are rather sensitive, but like our knees aren't, right? Our thighs aren't really sensitive. And that all, by the way, has neurophysiological representation in the somatosensory cortex, which is really cool. We can talk about that if you want. But anyways, and so using the tongue, this device, that palette, that grid of electrodes is hooked up to a pair of glasses that has a camera on it. And as the camera, is is exposed to visual scenes, the grid on the tongue delivers subtle, very, very subtle stimulation. And it takes a long time. It takes months and months of practice, but eventually people who are blind trained with it and were capable of using that camera as and the thing on their tongue as a way to navigate, as a way to move through a room and experience walls. And again, this is all through touch. And they would report that they even have visual experiences, right? So, the sensory nervous system is really, really amazing in how adaptive it is. And so part of whomever brought it up that was right, that like you're just not as distracted by all of this being bombarded with visual information, but it's also that the brain adapts and can enable sensory substitution. It's really cool. So I was first to mention Brain on Fire. You know about it now, right? Oh yeah. I watched it a long time ago. Yeah, it's an interesting movie. It's tragic in some ways, but it's worth checking out. You can hyperfocus on sound. Oh yeah, so those Don't Stop Believing. Yeah, pretty much. Oh right, let's do this. Okay, sorry I've missed comments on YouTube. Hey Ron, dude, I was just thinking about you, man. That is wild. Well, it was you where we talked about puppets, right? Am I crazy? If I'm crazy, then forgive me. But my daughter loves Sesame Street. She is like an Elmo aficionado, and so I was just reminded, because she's very into it and potentially very into puppets, and just reminded me of you emphasizing that they're very good educational tools, and you are right. You are 100% right. Okay, Keyboard Warrior. Will the vaccine be effective again? Oh, the proteins have passed the blood-brain barrier. Will the vaccine be effective? Okay, BBB. Sorry about that, Keyboard Warrior. I'm sorry, I was delinquent there. Good afternoon. Pleasure to see you here as always. Likewise, Craig. Thank you. I have a question with regards to the vaccines. I hope you could clarify for me. Do any of them prevent the spread of COVID, or do they make us asymptomatic carriers, right? What are the 92% effective in, right? Protecting us from mild symptoms. I'm on the ledge whether or not to get vaccinated. Thank you so much, but I'm sorry to get off topic. No, you don't have to apologize. Yes, you may call in, please. The short answer is we don't know for sure, okay? There is evidence that I believe it's the Pfizer vaccine does indeed reduce viral load in the nasopharynx. It doesn't eliminate viral load, but the end point of all of these vaccines is to reduce the severity of symptoms. And they're very, very, very effective at doing that. Unfortunately, hey, what's up, Euro? Good timing. Unfortunately, it looks like the vaccine that's produced in China, what is it called? It's like Sinovac or something like that, does not appear to be particularly effective, which is unfortunate because it is the traditional strategy of inactivated viral particles, which does theoretically have some advantages to it. And that's, I think, also true for a vaccine that's being produced in India. But we don't know the efficacy of that one yet, at least as far as I'm aware. All right, it looks like we're connected. Layback Dre. Yeah, good to talk to you again. Where are you calling from?
Miami.
Right on. So what's on your mind, man?
I wanted to know what causes intrusive thoughts and how can you get rid of them?
Sure. So intrusive thoughts. Would you do us a favor and describe like an example of intrusive thoughts?
Like, for example, like sometimes, like I think I go through intrusive thoughts. I'm not sure. Like sometimes I'll I have thoughts of positive stuff, but then again, I have thoughts of like negative stuff. Like, for example, I almost got hit by a car. That didn't literally happen, but it just went through my mind. Like, I think it's caused by like anxiety or something like that. Like, what if this happened or what if that happened?
Yeah, I mean, you're right. It is absolutely associated with anxiety. So, intrusive thoughts can be a symptom of actually obsessive compulsive disorder, where it can be, which is an anxiety associated condition. And I think most people, when they think of OCD, which is obsessive compulsive disorder, they think of the rituals, right? The turning the lights on and off multiple times, needing to arrange everything just so. Although that can also be the obsessive compulsive personality disorder. But anyways, the excessive hand washing, these types of things. But OCD can also manifest in ways that don't really have much to do with rituals. That are, it's rumination. It's being unable to stop thinking about a specific topic, generally speaking, unpleasant. And so, that is so part of it is that we evolved to really bias our cognition towards and our attention towards negative topics, right? Negative events, negative experiences. We respond more severely to negative emotional events. And I mean, I think that's pretty overtly demonstrated on social media, which is why like we're just bombarded with like this chaos, this horrible tragedy, right? Because it tickles our brain's erogenous zones for attention. And so, so that's just the sort of baseline of the human primate brain, right? But the second is that there is a brain region or a distribution of regions that regulates whether or not you become consciously aware of a thought, right? Our conscious awareness is just a small portion of our broader, brain physiology, right? In other words, like you're unaware of a lot of the brain activity that's occurring that then ultimately will serve up to you a conscious thought, right? Like, for example, if I were to ask you for your favorite food, right, you might say, I don't know, like a Cuban sandwich. And, you might think about it for a little while. You might say Cuban sandwich because you're in Miami and Cuban sandwiches are awesome. And in your mind, you would think, I made that decision, right? Like I made that choice. But really what was happening is that there was a bunch of subconscious processes under the hood that you could not see that then competed essentially with each other and then were sort of selected or bubbled up to the top by a part of the brain called, well, let's say two parts of the brain, one called the anterior cingulate cortex and the other, the whole striatum really. And so that's sort of the process. And so when it comes to intrusive thoughts, it's sort of a combination of those things, right? Where that, there's the fact that we're predisposed to these types of things, but then also that system, and you're right that it becomes it's altered when somebody's anxious, when somebody's pathologically anxious. That system is it's not operating properly, and therefore you recurrently are stuck in this loop of focusing in on something negative, something frightening, something anxiogenic. So that's fundamentally, and so how can you stop it? Well, I mean, the simple response would be, consult with a psychotherapist or a psychiatrist or whatever, particularly if it's like affecting your life, right? If it's like you're not able to live a normal life, which, totally can happen. But, alternatively, it's like if you can reduce levels of anxiety by doing a bunch of different things, making sure you get enough sleep, making sure you have a good diet, making sure you exercise, making sure, you remove sources, to the best of your ability, sources of pathological anxiety from your life, like people who are traumatic or whatever, that may yield benefits. But certainly what I would do is talk to a psychotherapist or a psychologist.
Okay. Thank you so much for your help, man. I appreciate it.
Yeah, thanks for calling in. Good talking.
All right, you too. Bye-bye.
I used to live in Miami, everybody. Was not my favorite city, but it has its positives. You're the one who introduced me to... No, you shut up. No, you. Okay, how do you feel about the Bill and Melinda Gates Foundation funding the development of genetically modified mosquitoes to become capable of human vaccination? I've not heard about that. That's very interesting. I find it interesting from just a purely scientific perspective, but I have not heard that that's true. There's been a lot of BS about the Bill and Melinda Gates Foundation, about vaccines in particular, and like, I'm sure some of it is authentic, is like authentic concern, and that the history of vaccines is not exclusively positive, right? But I think a lot of it is just intentional, malicious activity. People just like intentionally lying to cause harm to other people because it's the only way that they can feel a source of relevance, that they'll be remembered somehow, that because they made it so that they convinced a lot of credulous people to believe that the thing that would save their life and the lives of their loved ones is actually going to kill them. I think that's what a lot of this is, and it's unfortunate. All right, it's from an article in Science Magazine. Well, wait a second. I mean, were they talking about a theory, like a concept, or are they literally talking about making mosquitoes that are engineered to express the spike protein? I can't even imagine how that would even work. Like, they would have to. Yeah, I don't know. That's super interesting, Craig. Hey, listen, if you can, I would love a link to it in the chat. That would be, I would really, really appreciate that, because that's really interesting. Thanks for bringing it up, sincerely. It's all due to mistrust in government, rightfully placed mistrust. Yeah, certainly. There's a part of that for sure. Okay. Literally talking about vaccines in general. Okay, I mean, sure, but I mean, I'm specifically talking about the mosquitoes thing, like genetically modified mosquitoes. But are you sure they weren't talking about genetically modified mosquitoes to reduce the spread of malaria? Because that definitely has been a project for a long time. I mean, malaria is like, if you were to enumerate like humanity's greatest foes, malaria would be one of them. Like, for sure, the number of people that die from malaria is gigantic, and the amount of net human suffering that's caused by malaria it's almost incomparable. I can't really think of something that's quite as close. So then I've definitely heard about that, but not about vaccinations for COVID from genetically modified mosquitoes. But again, if you can find that article again, please post it. I will read it with bells on. Like, seriously. Thank you. Your response was great. Oh, thank you. So right. My friend has had her life saved by the Brain on Fire book. Doctors referred her to psychiatrists. That's fabulous. That's great. I requested. Okay. Is one company better than the others, right, when it comes to the vaccines? It's an interesting question. I mean, like, if there seem to be subtle differences in the efficacies of the various I assume you're talking about vaccines, but they're all really quite high, the efficacies. Like the Oxford AstraZeneca vaccine is quite high efficacy. Same with the Moderna, same with the Pfizer. I'm hoping that the Johnson & Johnson vaccine is equally as effective, because that one, it's not an mRNA-based vaccine. It is also a modified adenoviral strategy like the Oxford AstraZeneca one, but their model is just a single vaccine. It can be stored in like your refrigerator if you wanted, and it can be stable for months. That would be a game changer, because it's really hard to coordinate two shots, right? Like, it just is, and the production has to go, need more production, and it's really hard to transport these things to, like, rural parts of the country that don't necessarily have negative 80 C freezers. So that's what I'm hoping for. I'm pretty sure we're going to see the results of their trial like pretty soon. But yeah, so the Oxford AstraZeneca one, the Moderna one, the Pfizer one, they all have pretty comparable efficacies. There was a little bit of like an interesting event with the Oxford AstraZeneca one, where by mistake half of their study population, or half of one of the groups got like half a dose, and but that ended up being like the far more efficacious strategy, which was very funny. But yeah, we're talking 90+ efficacy, which is gigantic. And to give you like a sense, I think I brought this up earlier, the vaccine that's being produced in China, I think it's called Sinovac, something like that. that efficacy is like just above 50%. And so that is the sort of threshold that both the FDA here in the United States and the WHO sort of set for, is this even worth it, right? And so that's unfortunate. But again, I believe that there's a group in India who's also making an inactivated viral particle vaccine, which is basically where you take viral particles, you inactivate them, by irradiating them, by heating them up or something like that. And the benefit of that strategy, theoretically, is that rather than just being exposed to the spike protein, your immune system is being exposed to the entirety of the viral particle. It's non-functional, but so you can develop antibodies against more than just the spike protein, right? So you can develop antibodies against the different glycoproteins or just different parts of the virus, which therefore theoretically, if there is a mutation in one of the strains, you may be more likely to be still have an effective immune response despite that mutation. But that said, we still think that excluding maybe one of the recent variants, that the mRNA ones are going to be just fine. And once again, as I always say, like the mRNA vaccines can be very rapidly changed. Same with the adenoviral ones. Oh, and thank you for the super hearts. I appreciate it, Sherry. Thanks very much. I hope all's well with you. It's great to see you. I used to go as John Zoidberg back in the day. Oh, that's awesome, man. Cool. Correct, but that would imply that you can get a mosquito to vaccinate malaria. You would theoretically get it to no. So it's not a malaria vaccine. It's killing mosquitoes. They're mosquitoes that will end up reducing the mosquito population. That's the strategy. It's not something specific against malaria. At least those are the projects that I've seen. So Abyss, we're connected. What's on your mind, man?
Hey, can you hear me?
Yes.
Oh, okay. Yeah, usually my Bluetooth headset kind of turns off when I connect in Periscope. Yeah, so I guess like we talked about the visual cortex and sort of like its intricacies, the fact that it's one of the most energy consuming parts of our brain. It got me thinking, like, because like right now we have artificial models that can actually do edge detection and color detection as much as even or more faster than our brain can. Yeah. And if you go through like all the pathway, like from the retina into the suprachiasmatic nucleus up to like the V1, V2, and like it seems like the amount of information or the deluge of information is one of the key factors that kind of sort of escalates the requirement to process such a vast amount of information. Given that we have to interact with our system. But do you think? I mean, this is probably like borders somehow like a sci-fi, sci-fi realm. But I'm kind of like curious because do you think like removing or at least like substantially reducing these different pathways, so our visual process can essentially recognize faces and objects, and in the near future that means like somehow sort of like exporting this or outsourcing this object recognition to a computer system perhaps, or that's far more able to detect objects than we could.
I love it. Yeah. 100%. So basically, correct me if I'm misunderstanding. So basically, given the fact that the visual cortex, the occipital lobe, is so gigantic, right, relative to other brain regions, right. and that it, of course, emerged through evolution, which is an imperfect process. It's a tinkerer, not right an architect or whatever. That obviously a lot of that connectivity is suboptimal, or at least it's not optimized. It could be more efficient, and since we are now developing computer systems that are capable of executing pretty complicated visual feats, that if we ended up being able to integrate some of those systems into our own nervous system, that would sort of free up some real estate within the brain that could then participate in other things, right? Is that basically what we're talking about?
Right. Also, we can clearly, I mean, it's far more easier to do like a sensory motor somatosensory as well as motor prosthesis, right? I mean, it's essentially like, you have to locate which part in the, the motor gyres or sensory gyres that you can, or sulcus, actually, not gyres, but just the sulcus areas that you can locate which parts control the arm and everything.
Yeah.
But it seems like optical augmentation, or at least like, the fact that using some kind of electrodes to essentially input visual components, it seems to be far more complicated, and rightfully so. And this is why, like, I don't see this thing happening anytime soon. But I feel like this can be more important, and especially like people that did not have, like, visual input since like growing up, or like lost their sights. And one of the reasons why I asked is that we know that some organisms, like especially birds, for example, I think crows and ravens have far more, I mean, not far more, but like, as sophisticated object detection. In fact, like they can even detect the physiognomy of a person.
Mm-hmm.
They actually did a research on that one. So, and they don't have a gyra and sulci like we do.
Right.
They pretty much have like a smooth brain, but they're able to do object differentiation as much as we can. And it seems like the visual part of, or at least the occipital lobe is engaging in a very energy demanding task, if that makes sense.
Yeah, totally, man. Yeah, well, I mean, so I should acknowledge, I don't know, maybe.
Are you there?
Yeah.
I'm here, but it looks like the video's frozen.
Oh, is it really?
Okay.
Oh, no.
Oh, good. I was just going to ask in the chat if other people saw that, but I see other people saying that now.
Huh.
Yeah.
It's too bad, because this is like some of my favorite topics that we're touching on.
That's why I thought you should join.
And I missed part of the broadcast, unfortunately.
Huh, okay.
Maybe we'll be able to catch it on replay.
Okay, well, why don't I let
perhaps.
you go, Abyss.
I mean, he just talked about the the visual system earlier on, before he jumped on the the COVID and the genetically engineered mosquitoes discussion.
Yeah, that's the part. I mean, basically, I missed everything because I've been in and out. Well, first I wasn't in, and then even when I was in, I was doing something else. But
Well, guys, let me let one of you guys go.
On the COVID side, so of course, I was very interested in that.
Oh, can you not hear me?
Okay.
Oh,
I missed a
no.
couple of topics, but
Okay.
Yeah.
my research is on COVID, so
Oh, my gosh.
You mentioned that one time.
yeah.
Okay, let's see if this... Oh, man. This is a disaster. Oh, no. Shoot. Okay. Well, yeah. Well, fortunately we have YouTube, right? Here, maybe I can just do this really quick. Can I even... Wow. This is... Okay. Alright, so here's what we're going to try. So I have about 15 more minutes that I can hang out. Sorry, I know on YouTube this is just brutally weird. So we'll do this. Hello? Anything. Go live. Okay. Well, it looks like we can't do two at a time, so that's too bad. Hi, everybody. My name is Mclaughlin. I have a PhD in neuroscience at the University of Pennsylvania, and I was just cut off because I tried to add two people chatting at the same time, two people with substantial experience in science, and we were talking about the prospect of augmenting our visual system using computer systems that are, frankly, superior, at least in some ways, and theoretically they will become superior in many ways. it reminded me, I actually saw an eagle today. Oh, Abyss, you're back. Yeah. I saw an eagle today flying over Philadelphia. It was really cool. But like, the reason I thought of that is their vision is, obviously superior to ours, at least as far as we're aware. We can't know for sure because we don't know what it's like to see like an eagle. But anyways, it's an interesting, okay, wait. So scientists know less about our brain than space, and they don't know squat about space. Well, a lot of people make a lot of hay about saying that we don't understand the brain, we don't understand consciousness, and that's just not quite true. It's like we understand a lot about consciousness. We don't, it's probably not a sufficient amount to satisfy a lot of people in an intuitive way, but there's no rule that says that neuroscience has to be intuitive, Okay, so can you briefly discuss how ketamine seems to work so quickly compared with other antidepressants? Yeah, awesome. I would love to be the fly on the wall in that conversation. Okay, and then we can talk about this and the psychobiome. Okay, awesome. So yeah, so ketamine. So first of all, we don't actually know exactly how ketamine is effective. We have some theories, but one of the theories is that essentially what it does is it amplifies the activity from the neocortex onto certain parts of the brain that regulate mood and emotion, and therefore depression. And it's an immediate effect. And one of the theories for how antidepressants like SSRIs, more classic antidepressants like SSRIs work, is that due to that chronic elevation of serotonin, because SSRIs are not immediately effective, as I'm sure you're bringing up. It is the chronic elevation of serotonin sustained for weeks, sometimes 2 or 3 weeks, that then causes a similar type of response in the relationship between the neocortex, or just excitatory glutamatergic neurons that release glutamate specifically, or neurons that have receptors for glutamate specifically, that delivers the therapeutic effect of SSRIs. And so what is an immediate effect for ketamine is a delayed effect for SSRIs. That's just one possible theory. It's not necessarily true. Makes sense. Totally makes sense. But that would be one reason. Another reason is that literally the pharmacology of SSRIs, selective serotonin reuptake inhibitors, we're talking about drugs like Prozac or fluoxetine, the pharmacological effect is very delayed. And I actually measured this in my own lab work, where it's not like MDMA, where like you give MDMA and then levels of serotonin just go right, and just explode. It's like it's this slow increase, slow and steady and sustained increase. So that takes a while, right? It's not this giant change in neurochemistry. Whereas with ketamine, it's an immediate effect. It's binding NMDA receptors, antagonizing NMDA receptors, and that has an immediate effect. So that would be some of distinctions. Am I a scientist? Yes, I'm a neuroscientist. I have a PhD in neuroscience. Hi, and this is a topic I would love to hear your thoughts on, and the psychobiome and epigenomics. But gotta get back to the NFL. Oh yeah, no worries, of course. Thank you very much. I appreciate the super heart. Regarding ketamine, a girl once asked if I gave her Special K, and I told her I don't eat cereal. Did you really though? Or did you really not know what she was talking about? Live calls are a bad idea. Scrolling and answering is better. Well, but it is and it isn't, because like having a sort of sustained conversation. I mean, I've been doing this for a long time, and having a sustained conversation and hearing the voice of somebody, I think, also is nutritious in unique ways, in ways that just messages don't. But I hear you. Hey, Natalie. Thanks for joining in. What's your job? So now I'm a science advisor to a state government. That's my main job. I had a second part of the question, but I'd rather DM because I need to find the publication first. Okay, yeah, that'd be cool. Are you optimistic blindness will be cured by bypassing the whole eye and jack right into with a computer? That is one potential treatment. Yeah. There are a variety of different treatments. So blindness can arise for a variety of reasons, right? Blindness can arise, again, from I guess not again in this stream, but damage to the retina, damage literally to the eyes, damage, some kind of intrusion upon the pathways between the eyes and the visual cortex. It can also be damage to the visual cortex itself. It can, if it is just it doesn't have to be damage, right? It can just be like some kind of unique aspect of a person's biology at birth. So if it's just damage to neurons, then potentially just, by regenerative medicine may be a superior way to do it, because it'll just be more sort of intuitive. It'll be more like normal, feel like normal, because you're just replacing lost neurons or connections between neurons. However, I mean, I think it is not a stretch to suggest that at some point, as technology improves, we will be able to, as Abyss was suggesting, integrate computer systems into the brain, and that would include, optics. What happened to Malaysia Flight 370? I don't know. It's a weird story, isn't it? How does the brain access memories? It's an interesting way to phrase that, B. Phillips. So, because of course the way you're phrasing it sort of suggests like the brain must be accessing memories the same way a computer accesses memories, right? And the way a computer accesses memories is that it's either stored on, a solid state drive, a hard drive. It's stored physically in a physical place, or whatever. It's in RAM for a sec, right? There are some analogies, but it's not quite the same. And so it's more similar to like, okay, first of all, there are different kinds of memory, right? So when it comes to a computer, memory is memory. It can store whatever kind of thing you want it to store it in the same way. It can be stored in the same place if it's substituted physically, right? Where you can store a text file, an image, it doesn't matter, movie, who cares? But when it comes to the brain, different brain regions underlie different forms of memory. So, I will spare everybody who's seen this before my classic neuroscience parlor trick of the steering wheel thing. But we have procedural memory, episodic memory, declarative memory, short-term memory, working memory, all of these different forms, and they're all encoded in different brain regions. But so let's just talk about, like episodic memory, right? So meaning like, do you remember where you were when you had your first kiss, right? Like that kind of thing, if you've had a first kiss. Or do you remember where you were when you heard your favorite song, or your first experience going to a concert or something like that? That's episodic memory, a personal autobiographical sort of thing. So that involves a bunch of different brain regions, most famously the hippocampus, and it'll involve, over time, however, essentially you can think of it sort of like, as you're having this experience right now, you're experiencing something visual, theoretically, if you can see your device. You're hearing things, auditory experiences. I'm bringing up words like your first kiss, your first concert, and so you're having memory experiences that are being just sort of tickled here and there. And as a result, different constellations of neurons are activating, right? Changing millisecond by millisecond. And the way the brain we don't know exactly how the brain stores memory, but one theory is basically there's a sort of compressed short-term version of it that is sort of inefficient but accessible, right? Like it just happened. You just remember that I said your first concert, right? But then over time that transitions to a different brain region that is sort of essentially like generating a compression file of the constellation of neurons that were activated. And then over time, that form of memory, shorter term memory, then migrates, at least in part, to the neocortex. And the human neocortex is actually just gigantic relative to other animals. Like we have more neurons in our neocortex than elephants do, despite the fact that elephants have more neurons than we have. Right, so we have this big fancy neocortex, and one of the things that it does is it enables longer term memory. But the takeaway, at least for me, is that human memory, that is also, by the way, before I close, that is also one of the reasons for why human memory is so fallible. Right, computers technically have superior memory to us because our memories are stored in biological wet stuff that are subject to change. Every single time you remember a thing, it actually enters into this state of vulnerability, this malleable state where things can change. Things can begin to change in your memory. Like, for example, I'm wearing a green sweater with these, lines on it, right? Over time, when you think about this crazy conversation you had with this nutty neuroscientist, looks kind of like that guy from Heroes. You might, was it a black sweater though? Or wait, was he in Philadelphia or was he in like Los Angeles or New York City or something, right? So like all these little aspects as a byproduct of that constellation of neurons being reactivated, or not the entire one, but a constellation of neurons being reactivated, proteins being synthesized, synapses being activated, removed, created, right? And so obviously polka dots, yeah, my favorite. And so that's part of the reason for why human memory is so fallible and inferior, frankly, to other forms of memory. But that said, it is obviously far more versatile, right? So yes, but so basically that's how humans access memories. We have sort of compression files of constellations of neurons that are activated for episodic memory at a given moment, and the substrate, you know, it moves from RAM to your hard drive, right? It's not quite the same, but the place, the physical space of that memory evolves over time. Great topic. Fun fact: hippocampus is also where hippopotami go to school. Very cute, Scott. Very, very cute. Can you train your brain to focus more? Yes, you can. And then what happens in the brain when we fall in love? Awesome topic. Let's talk about that next. So first of all, a lot of human behavior is subject to environmental influence, and so like self-control, for example, I think is my favorite, because like, at least of the studies I've seen, it has the most overt or obvious demonstration where you know, self-control, we'll just sidestep the whole philosophical conversation about whether or not self-control is even a thing, free will is even a thing. Let's just pretend like that question doesn't exist for a second. Self-control emerges from, again, a whole network of different brain regions that involve more primitive parts of the brain, right? Certainly excited neurons. Some of the neurons are certainly excited, but some of them are inhibited. Some of them are depressed, relaxed. Certain parts of the brain that happen to be evolutionarily more primitive, that have been around in an animal of some kind for hundreds of thousands, millions of years. Those subserve our urges, our gut sort of visceral desires, our libido, our appetite, our desire for or draw to hedonistic sort of pleasure, right? And so those brain regions are older, part of the brain regions that I studied, by the way. And then humans uniquely have this big fancy prefrontal cortex, really just frontal cortex. Other primates have a prefrontal cortices, but the human prefrontal cortex and frontal lobes are significantly more robust and just bigger than any other animals. And so why does that matter? And thank you very much for the super hearts. Why does that matter? What does the prefrontal cortex do? It does a lot of things, but relevant to this topic, it sends inhibitory off signaling neurotransmitters, or I should just say signals, to those primitive regions of the brain that drive urges and those types of primitive drives. And so the prefrontal cortex also receives all of this information, these signals from your past experiences, your mood, your things you've learned, these types of things, and can then inhibit those urges. So in the moment where you're like, man, do I really want to eat a second whole pizza, right? Or do I really want to eat another half a pint of ice cream? In the moment where you're like, no, I mean, I do want to eat it, but it's not good for me, I shouldn't do it. In that moment, what's happening in part is neurons in the prefrontal cortex are being excited and then send inhibitory off signals to those primitive parts of the brain. And the point being, can you train yourself to focus? Can you train yourself to exercise better self-control? Every time you do that, every time you exercise self-control, there is evidence that connectivity, that functional connectivity, that ability of the prefrontal cortex to inhibit those primitive parts of the brain gets greater, is hardened, is emphasized, is strengthened. So just like that, one's ability to focus. I mean, I'm happy to go on and on about focus. Like, focus emerges from actually a dopaminergic pathway, the mesocortical limbic dopaminergic pathway, and that is also similarly will be subject to sort of what your brain, what that circuit has determined as a suitable, acceptable level duration of focus for any given task. So there's, and that threshold can, well, theoretically, right, should be subject to change. I can tell you one of the ways that I did it when I was in college, and I had to read through a bunch of, or when I was in grad school particularly, when I had to read through a bunch of, genetic regulation classes, memorizing transcription factors and all this horribly boring stuff. I had to get really emotional about it. Like, I had to be like, if I don't pass this class, I'm not going to get my PhD, and I'm not going to be able to have a kid, or I'm not going to be able to provide for my kid in the way that I would like to. And so I really need to learn about CD8 plus, T cells, right? And literally, like, in my mind, what that's doing is it's just cranking up that dopamine signal just a little bit more, because the perceived reward at the end of this otherwise brutally boring task was greater, right? Because I made it a tangible reward. So, long response, I know. I apologize for that, everybody. Delayed gratification, my biggest foe, indeed. Yeah. All right, I should probably call it a day, everybody. So I have a YouTube channel. YouTube.com/anthropoid. Today I talked about the psychobiome, the relationship between the trillions of microbes that inhabit your body and the state of your brain, and how that influences predispositions to psychiatric conditions and psychology in general. So youtube.com/anthropoid. You can find my channel on my profile page on Periscope. I'm also on a new live streaming platform, which I usually do on during the weekdays. Called HAPS, HAPPS.TV. It's a great platform, really great community. It's still growing. It's very, very new. It's still growing, and it has a lot more tools than Periscope has. And so you can find HAPS, HAPPS.TV, by going to my profile page again on Periscope. And I can't remember which one's on top. I think HAPS or YouTube. There's two links. One of them is my YouTube channel. The other one is my HAPS. So, does HAPS have a chat feature? Yes, it does. Yeah. It's pretty good. And it has like call-in features and stuff like that are frankly a little bit more robust than Periscope's. Obviously, it's not directly integrated into Twitter, but yeah. Other than that, thanks for hanging out, everybody. And I hope you all have a healthy, safe beginning of your week. And hope to talk to you again. Same time, same place, next week. See you there. And all right, thanks everybody for hanging out on YouTube. I appreciate it, and thanks for being chatty. It's really fun that way. Yeah, your Twitter feed died, I know. But where is the index that sends the correct connection to the memory? Right? How do we generate the potential energy that turns into actual transmutation or transmission? Well, so where is the in other words, how is it that the correct constellation of neurons is activated, right? That it's an interesting question. I don't know that we have an answer to that quite yet. I could speculate, but a lot of it is going to be associated with there just being like physiological connectivity between, okay, so let's just play around with this topic for the last topic. So I say the word. I say the word sushi. Okay. I don't know if you've ever had sushi, but you probably know what it is, right? And maybe you've been to Japan. Maybe your favorite restaurant in the world is sushi. Maybe you went to a sushi restaurant and you got food poisoning. You had a terrible experience, right? You have all of these associations with the word sushi. So what happened is that I said the word, right? Obviously, the auditory system was activated, and you had to decipher all the sounds, all the patterns of the sounds that I make with my face, right? And as your auditory system is extracting meaning from that word, that process of extracting meaning then inevitably activates related stored experiences, right? Sushi, the word, the meaning of the word is associated with that experience, because when you're having that past experience, you also heard the word sushi or thought the word sushi, right? And so there's just this inevitable activation via the auditory stimulation. That's just one example, but it would be sort of something like that. I would think that it's just this cascade of what can be an extraordinarily complicated network of neurons, but it is just this cascade of this neuron activating this one, inevitably activating this one, and then you think of the circus, right? Okay. So yeah, thanks again. If you haven't hit the subscribe button and you've been hanging out this whole time, maybe give it a little prod there. And otherwise, please like it if you do like it. Don't like it if you don't like it. And so yeah, otherwise I will see you next week. Thanks everybody.